The liver is organized into blocks of tissue known as lobules. These lobules often have geometric shapes, often hexagonal, although there are differently shaped lobules. And blood flows from two different sources, branches of the hepatic arteries and hepatic portal veins. These two sources of blood fuse, forming sinusoid capillaries, which flow through these liver lobules. Sinusoid capillaries are the type of capillary with the largest spaces between endothelial cells, and thus are the most porous, allowing not only greater amounts of diffusion, but even proteins the opportunity to enter and leave the blood at these sites. Given the large spaces between endothelial cells in the sinusoid capillaries, the liver cells, the hepatocytes, are thus able to produce proteins which enter the blood, such as the albumins and globulins, which help to determine the osmotic pressure. When water leaves a capillary due to blood pressure, it returns due to the blood colloid osmotic pressure. Water goes back into the capillaries because of the colloids there, the protein synthesized by the liver. Many clotting factors are synthesized in the liver and enter the bloodstream through these sinusoid capillaries. Even proteins involved in the immune system, such as the complement proteins, which assemble when antibodies have bound an antigen and generate a pore in microbial membranes. These complement proteins are synthesized by the liver and enter blood in the sinusoid capillaries. The hepatic portal vein brings deoxygenated blood with remains of meals to the liver, and the liver performs many different processes with items from the diet. For example, monosaccharides can be stored as glycogen. Glycogen can later be broken down in the post-absorptive state to raise blood glucose levels. Glucose can be stored as triglycerides, as can amino acids. Amino acids can be synthesized from other molecules or one converted to another. Glucose can be made from other items in foodstuffs. And then harmful elements of the diet can be broken down. Hormones, whether they be from an individual's own blood or ingested hormones, can be broken down. Heavy metals can be bound. Alcohol can be broken down with alcohol dehydrogenase. Thus, there are a great diversity of molecules entering and leaving these sinusoid capillaries as the liver performs its very diverse functions. The liver possesses monocytes, known as Kupffer cells, or the liver's mononuclear phagocyte system, which can undergo phagocytosis of many things that might be in the blood, ranging from microbes to old red blood cells, many of which are processed in the liver so that they do not burst in circulation. While the liver can store triglycerides and break them down into ketone bodies in the post-absorptive state, in certain conditions, such as obesity and diabetes, the liver can store excessive amounts of adipose as adipocytes accumulate in fatty liver disease. Liver cells, or hepatocytes, are interesting cells. Very often they have more than the typical number of chromosomes. They can be tetraploid with 4 copies of each chromosome. They can even be octoploid. They may possess more than one nucleus, and the types of mitosis that liver cells can undergo is different from the typical mitoses throughout most tissues.